Hello dear students, welcome to our biology program. Today we are going to study molecular biology and especially the molecular technology. Let's begin. Molecular technology: how genes are turned on and off for the production of proteins. We still have much to learn about that. Our knowledge about genes has already given rise to a whole new technology called genetic engineering. From the achievement of this technology that it is possible to: Number 1, isolate a desired gene and grow millions of copies of it in the cells of bacteria or yeast. Analyze the copies of genes to determine the sequence of nucleotides. Compare the structure of different genes in the same or different organisms. Number 2, once we have found the sequence of nucleotide, we can determine the sequence of amino acids in the corresponding protein. Number 3, transfer functioning genes into the cells of plant or animals. And finally, make DNA by order, artificial gene. In 1979, Quran produced an artificial gene. This gene it was introduced in 1979. Quran produced an artificial gene and introduced it into a laboratory culture of bacteria. Many laboratories are now equipped with gene machines that can be programmed to produce short strands of DNA in any desired sequence. Tailor-made DNA can be used in protein synthesis experiments by changing the code for particular amino acids from protein. Tailor-made DNA can be used in protein synthesis experiments by changing the code for particular amino acids from a protein. Biochemistry can determine how amino acids affect the function of the protein as a whole. Some genetic engineering techniques. Now, the genetic engineering, we are going to study some of these techniques. First, nucleic acid hybridization. If the molecule of DNA is heated to 100 degrees, the weak hydrogen bonds between the two nitrogenous bases will be broken, and the two strands of the double helix separate to produce single-stranded DNA, which is unstable. When the temperature is lowered, it tends to stabilize by annealing to another single strand to form a double helix once again. For a notice for us, the degree of annealing between any two nucleotide strands can be measured by the heat required to separate them again. The more tightly the strands are bonded together, the more heat will be required to separate them. Any two single-stranded of DNA or RNA can form double strands provided they have at least two short complementary base sequences. The ability of single-stranded DNA and RNA to anneal can be used to produce hybrid or mixed double helix. The production of hybrid DNA mixed double helix. The means of hybrid here, that means it is a mixed helix or a mixed DNA. and we are going to understand what is meant by mixing. Pay attention and you will follow me, and you will understand. Mixing nucleic acid from two different sources, for instance from two different species of organisms, and heating them to 100 degrees. When the mixture is allowed to cool, some of the original helix will reform, but any new hybrid double-strand helix will form, each made up of one strand from each source. Uses of nucleic acids hybridization. First, for determining evolutionary relationship between different species. The closer the evolutionary relationship between the two species, the more similar their DNA sequence should be, and therefore the greater the degree of hybridization between them. Detect the presence of particular gene, and in what amount. Preparation of a single-stranded sequence of nucleotide complementary to one strand of the gene is in question is prepared using radioactive nucleotide so that the sequence is labeled and can be identified later. Mixing the radioactive strand with the unknown sample, the concentration of the gene in the sample is indicated by the rate of the formation of radioactive double helix. A radioactive double helix consists of one artificial radioactive strand and one strand of the gene in the unknown sample. So from this we knew that the nucleic acid hybridization, it means that the DNA, when it is heated to 100 degrees, so the hydrogen bond between the two helix will be broken, and then if they are cooled, so they are going back again and they complement each other, forming a double helix. From this one, the scientists make use of them by annealing the two different strands, but they have the complementary nitrogen bases for each other. So they begins by heating two different species of living organism heating them till the 100 degree, so the helix for each one, or the DNA for each one of them, will be separated into single strands. Then once they are cooled, the helix will the strands come together again to complement each other. Then we found that one of the strand, it comes from the first species, and the other strand, it comes from the second species, forming the double helix, and at this moment it is called hybrid helix. And then we are going to take another point, which is called bacteria restriction enzymes. They are enzymes which are specialized for making some cuts into the DNA. Phases of infection of bacteria. It was known that the virus invades certain strains of E. coli, which are restricted to these strains and cannot grow inside other strains of these bacteria. It was found in the 70s that the resistance of bacteria produces enzymes that attack specific nucleotide sequence in the viral DNA, and and break the DNA into useless fragments. These enzymes are called restriction endonuclease. So this restriction endonuclease, it is an enzyme which is make a cut into a certain sequence of, or breaks the certain sequence into the DNA. The question now is why the enzyme don't destroy the bacterium's own DNA. The question is why this enzyme restriction endonuclease doesn't destroy the bacterium own DNA or the DNA of this bacteria. Because each species of bacteria has modification enzymes which recognize the same DNA sequences that are attacked by its restriction enzyme and attach methyl group CH3 to same recognition site. This group of CH3 protects the DNA by blocking the binding of restriction enzyme to it. I'm going to repeat it because it is a very important one, and also it is a very important question. Why the enzymes don't destroy the bacterium's own DNA? This is because each species of bacteria has modification enzymes which recognize the same DNA sequences that are attacked by its restriction enzyme, attach methyl group CH3 to same recognition site. These groups of CH3 protect the DNA by blocking the binding of restriction enzyme to it. So this group of amino CH3, this group of CH3 protect the restriction and block the binding of restriction enzyme to it, to this DNA. The properties of the restriction endonuclease. Spread in microorganisms, especially in bacteria. More than 250 restriction enzymes were isolated from different bacteria strains. Each enzyme recognizes a specific sequence of from 4 to 7 nucleotides. 4 to 7 nucleotides in DNA, and snips the DNA at or near this recognition site. The sequence of bases on the two strands of DNA at the recognition site are the same when each is read in the 5' to 3' direction. So each enzyme recognizes a specific sequence of 4 to 7 nucleotides of DNA and cut them near the recognition site. Each restriction enzyme will cleave DNA from any source, such as viral, bacteria, plant, or animal, as long as that DNA contains one or more copies of its specific recognition site. Uses of restriction endonuclease. What are the uses of restriction endonuclease? It is a way to break DNA into pieces with known nucleotide at their ends. To break DNA into pieces with known nucleotide at their ends to produce many species with staggered cuts, because the piece of the pieces of the double helix are left with two single-stranded ends. These single-stranded ends are called sticky ends because they will base pair with any other single strand and produced by the same restriction endonuclease. So there is a recognition site and the restriction endonuclease break down this DNA at this recognition site, and this one left the strand cut it after being cut with two ends which are sticky ends. We call them sticky ends because they are single-stranded and they are needed to base pair with another single-stranded. But there is a condition here which is very important, that the second single strand must be produced from the same restriction endonuclease. Splicing of a specific section of DNA, a gene, into another DNA molecule or plasmid. Breaking DNA into pieces with known nucleotide at their ends by the same restriction enzyme. The cut ends can be joined into a single-stranded by DNA ligase. So we are going to repeat the same part because this part will continue with us in next time when we are going to study the next session, the cloning one. We can use restriction enzyme to produce a single-stranded or to cut the piece of DNA. So the DNA, it is cut into pieces with known nucleotide at their ends to produce many pieces with staggered cuts because the pieces of the double helix are left with two single-stranded ends. So this one, the restriction enzyme, works at a certain site with a which we call it recognition site, and the cut it is done at this part. This due to the sequence of some nucleotides from 4 to 7 nucleotides. If they are found, if the restriction enzyme found them, so it begins to make a cut at this part or snip it. Then after that, the DNA at this part, it will be single strand because the hydrogen bonds here, it will be cut into at this part or break down. Then after that, the two ends are, we call them sticky ends because they will base pair with any other single-stranded ends, and they produced a new one. But there is a condition here which is very, very important, that this new strand which is sticked to this or base paired with this sticky ends must be produced from the same restriction endonuclease. Then the splicing of species of specific section of DNA into another DNA molecule or plasmid. Breaking DNA into pieces with known nucleotide at their ends by the same restriction enzyme. Cut ends can be joined into a single strand by DNA ligase. Now, my dear students, we came to the final part of our program, the questions. First question. The first question. Choose the correct answer. Enzymes attack specific nucleotides sequence of the viral DNA. Ligase, endonuclease, lipase, insulin. Yes, endonuclease enzymes attack specific nucleotide sequence of the viral DNA. Each restriction endonuclease enzyme can recognize a specific sequence of space nucleotides 4 to 5, 7 to 9, 5 to 9, 4 to 7. Yes, 4 to 7. Second question. Give reason for the restriction endonuclease of a bacteria do not destroy its own DNA molecule. The restriction endonuclease of bacteria do not destroy its own DNA molecule because each species of bacteria has modification enzymes which recognize the same DNA sequences that are attacked by its restriction enzyme and attach methyl groups, which are CH3, to the same recognition site. These groups of CH3 protect the DNA by blocking the binding of restriction enzyme to it. Give reason for. We can determine the degree of annealing during DNA hybridization by using the heat, because if the amount of heat that is required for annealing the hybrid DNA is low, this indicates that the degree of annealing is low and vice versa. We can determine the degree of annealing during DNA hybridization by using the heat, because if the amount of heat that is required for annealing the hybrid DNA is low, this indicates that the degree of annealing is low and vice versa. This is a very important question. Number 3. If DNA molecule is treated by restriction enzyme, the ends of cut pieces are called sticky ends. Give reason. If the DNA molecule is treated by restriction enzyme, the ends of cut pieces are called sticky ends. The ends are called sticky ends because the nitrogen bases of each end can be paired with other single-stranded end, which is produced by the same restriction enzyme. The fourth question, give reason. DNA hybridization can be used to determine the evolutionary relationship, because if the evolutionary relationship between two different species is closer, the degree of hybridization between them will be greater, because the sequence of their DNA is more similar. I will repeat this question another time. DNA hybridization can be used to determine the evolutionary relationship. Yes, because if the evolutionary relationships between two different species is closer, the degree of hybridization between them will be greater, because the sequence of their DNA is more similar. The third question. You have to be careful when answering the questions of right or wrong, because sometimes there is some tricks in this question. It's a tricky question. Then put right in front the correct statement and wrong in front of the wrong statement. Then correct the wrong ones. The restriction endonuclease are spread in virus. Wrong. Bacterium strands. Bacteria strands. The correction is a bacteria strands. Each restriction endonuclease recognizes a specific sequence of 4 to 9 nucleotides. It is wrong also, and the correction is 4 to 7 nucleotides. The third question: hybrid DNA molecule is formed of 2 single strands, have short complementary base sequence. This is right. Hybrid DNA molecule is formed of 2 single strands, have short complementary base sequence. Fourth question: what are the importance of molecular technology? The importance of this molecular technology is, first of all, it isolates a desired gene, analyze the copies, compare the structure of different genes. So it is very important because it isolates a desired gene and grow millions of copies of it in the cell of bacteria or yeast. Then analyze the copies of genes and determine the sequence of nucleotide. Then compare the structure of different genes in the same or different organisms. Number 2, determine the sequence of amino acids in the corresponding protein. Number 3, as an importance for this molecular technology studies, transfer the functioning genes into the cells of plants or animals. Transfer functioning genes into the cells of plants and animals. Make DNA by order as an artificial gene, as Quran in 1979 produced an artificial gene, and also tailor-made DNA which can be used in protein synthesis experiments. The fifth question: mention the uses of each of the following. Number 1, restriction endonuclease enzyme. Okay, break DNA into pieces with known nucleotides at their ends that are called staggered cuts, because the pieces of the double helix are left with 2 single stranded ends. Second importance or second use: splice a specific section of DNA, a gene, into another DNA molecule or plasmid by DNA ligase. So the uses of restriction endonuclease enzyme is break down into pieces break DNA into pieces with known nucleotide at their ends that are called staggered cuts, because the pieces of the double helix are left with 2 single-stranded ends. Splice the specific section of DNA, a gene, into another DNA molecule or plasmid by DNA ligase. The second, mention the uses of hybridization. Determine the first determine evolutionary relationship between different species. The closer the evolutionary relationship between the two species, the more similar their DNA sequence should be, and therefore the greater the degree of hybridization between them. Number 2, detect the presence of a particular gene and in what amount. Preparation of a single-stranded sequence of nucleotide complementary to one strand of the gene in question is prepared. Using radioactive nucleotides so that the sequence is labeled and can be identified later. Mixing the radioactive strands with the unknown sample, the concentration of the gene in the sample is indicated by the rate of formation of radioactive double helix. Radioactive double helix consists of one artificial radioactive strand and one strand of gene in the unknown sample. A radioactive double helix consists of one artificial radioactive strand and one strand of gene in the unknown sample. And now, my dear students, we reached the final point of our lesson. See you next time.